Hi, welcome to this next video tutorial in our general biology course. This session is entitled Cell Division Part 1. One of the fascinating things that life can do is produce more life, and that all begins at the level of cell division. So let's take a look at our learning objectives. We want to talk about cell division and the cell theory, the process by which cells produce more cells. One of the ways cells do that is by binary fission. We'll see that's how bacteria cells do it. And then we'll need to talk about some chromosome terminology if we're going to keep track of things as we discuss eukaryotic cells. And then we'll want to look at the process by which eukaryotic cells divide, and that's called mitosis. So that's what I want to try to get through in this first video lecture. So again, all semester long we've been asking this question, what is life? And of course, we've come up with our list of 6 things. We've worked our way through several of these things, and we're now moving into this idea of growth and development. Living things grow and change and multiply and so forth, very different from rocks. So for example, these frog tadpoles growing in their eggs started out very, very small, but through cell division, and not just cell division, but also cell differentiation, producing different types of cells, these tadpoles are going to eventually become adult frogs. So life has come up with a way by which cells can become more cells and grow and go through development. Now that should take us back to the cell theory. Remember, the cell theory had 3 statements to it. All living things are made up of cells. Cells are the smallest units of life. And then the third statement, which was largely put forth by Rudolf Virchow, was the idea of omnicellula e cellula, every cell from a cell. This is the statement that all cells come from pre-existing cells. Cells don't just arise spontaneously. So he's the one we can credit with that piece of the cell theory. And if we're going to talk about how cells come from cells, how life begets life, it's probably best to start by looking at how that works in bacteria cells, in prokaryotic cells. So just to remind you, here is our poster child bacteria cell. Remember that bacteria are prokaryotic. Prokaryotic meant coming before a nucleus. So they do not have a nucleus. They do have a chromosome in there. You can see it, that tangled up purple stringy stuff. That is their chromosome. It's actually one big loop of DNA. And when they go through cell division, they're going to have to copy that chromosome because that's the genetic material. And then they'll simply split in half. The process by which prokaryotic cells or bacteria cells divide is called binary fission. And you can see some bacteria dividing through the microscope here. 1 cell becomes 2, 2 become 4, 4 become 8, and 8 become 16, and so forth. So they're dividing very, very rapidly. The word binary fission literally means splitting in 2. Binary means 2. Fission means to split. Fission is the opposite of fusion. So binary fission literally means splitting in 2. And this is going to be the process by which prokaryotic cells multiply. When they do this, the resulting cells, which we call daughter cells, are going to be genetic clones of the cell that divides. So we're not going to create any new genetic change or anything like that. This is just one cell becoming 2 cells that are identical to that cell. And if they do this, they can double the population about every 30 minutes. As you watch them divide under the microscope, you can see the population doubling and then doubling and then doubling. Now I will say that what I put under the microscope there was time-lapsed, so they were doubling faster than every 30 minutes, obviously. But bacteria can divide about every 30 minutes, and that means the population could double every 30 minutes. All right, so here's how binary fission works. You start with the bacteria cell, and you can see it has its chromosome in there. That's the genetic information, the instructions on how to do everything in the cell. And so the first step, if we're going to turn this into 2 cells, is that genetic information, that chromosome, has to be copied. So we're going to learn the specifics of that process several videos in the future. It's called replication. But basically, the prokaryotic chromosome gets duplicated. So then there are now 2 identical chromosomes in that bacteria cell. Once they have the genetic information duplicated or copied, it's really as simple as just starting to split the cell in half down the middle And again, you get two daughter cells. So that very first cell at the top is referred to as a parent cell, and then it splits to become two daughter cells. And those two daughter cells are completely identical, clones of each other. And moreover, they're both clones of the original parent cell. So that's binary fission. It's actually pretty straightforward. Take your chromosome, copy it, split down the middle, and ensure that a chromosome ends up in each daughter cell. So that's prokaryotic cells. But what about eukaryotic cells? Remember the type of cells that fungi and plants and protozoans and animals, including humans, are made of. So here is our poster child eukaryotic cell. And the process by which eukaryotic cells divide is called mitosis. Now, it's a little different. In some ways, it's actually very similar to binary fission. Copy your chromosome, split in half, make sure you get the same chromosomes in each daughter cell. But eukaryotic cells usually have multiple chromosomes, and that makes the task just a little bit more complicated. In fact, we don't even see them as chromosomes at this point. You see this label chromatin. Inside the nucleus is chromatin. That means colored material. The chromatin is a coiled, tangled up collection of all of the chromosomes. So if you think of a chromosome as a DNA molecule, your cells, for example, have 46 of these chromosomes, 46 DNA molecules, and normally in the cell they're all just tangled up into one big mess that we call the chromatin, the colored materials. And because the chromosomes are all tangled up with each other like this, it makes the whole process of getting them copied and splitting the cell in half and getting the exact same kinds of chromosomes in each daughter cell a bit more complicated. But let's look at it simplistically first. In fact, let's just deal with one chromosome. Again, remember your cells, for example, have 46. But let's just think of one chromosome for a second and see what goes on during mitosis. So this could be one of those chromosomes, one of the DNA molecules that's in that tangled pile of chromatin. So if we want a definition, a chromosome is an individual DNA molecule, and as I've said a couple times now, humans have 46. Don't let the total number of chromosomes mean a whole lot. Fruit flies have 4. Goldfish, I believe, have 95, so it's kind of an arbitrary number, but we have 46 of these things. Another way to draw that would be to sort of show it literally as a DNA molecule, which is what they're showing here. Now, much like binary fission, the first step is going to be to copy the chromosome. So we see chromosome duplication occurring. So now we have 2 copies of it, and those 2 copies are referred to as sister chromatids. So sister chromatids are duplicate copies of a chromosome. So we're getting a lot of C words here. We got chromatin, chromosome, and chromatids. So be sure you're keeping track of these different terms. And we could draw those like this. So here are 2 duplicate copies of the same chromosome held together. The chromosome existing as 2 sister chromatids can even be seen under the electron microscope. So that's what you see here. You can see the 2 sister chromatids held together. We would refer to that as a single chromosome still, one of the 46 chromosomes. It just happens to exist at the moment as 2 duplicate sister chromatids. And they're held together by a structure called the centromere, so yet another C word. The centromere is the region where sister chromatids of a chromosome are joined together. Now, once the sister chromatids are joined there by the centromere, what the cell's going to do is throw some ropes out and pull them apart from each other. So we're going to pull those chromatids towards opposite sides of the cell. And what that will do is ensure that when the cell divides, each daughter cell gets a copy of that particular chromosome. The word mitosis, the mito, literally means thread. So ropes or threads are thrown out to the sister chromatids, and then a game of tug of war is played, and they're pulled apart. So it's really the thread process is what the word mitosis literally means. And again, we could see 2 DNA molecules, 2 duplicate copies of the same chromosome have been pulled apart, and then when the cell splits in half, hopefully one ends up in each daughter cell. So we just showed what happens to one chromosome, but you need to be thinking about that process taking place with many chromosomes. So let's look at the process of mitosis in a little bit more detail and see what goes on inside the cell. So here is a cell, and you can see inside the nucleus the chromatin, the tangled mess of all the chromosomes in just one big super coiled up pile. So there's the cell that has not begun mitosis yet. In fact, when a cell is not in mitosis, we say it is an interphase, the word you see at the top, so interphase. So most of the cells in your body right now are in interphase. They're just doing their normal cellular activity. But at some point, the cell will know it needs to divide, and so the first step is to get the chromosomes copied, and technically that occurs while the cell is still in interphase. So it actually occurs prior to mitosis beginning. So the chromosomes get duplicated. So now in that tangled mess of chromatin, we should see twice the amount because every chromosome in there gets duplicated to exist as sister chromatids. The cell is now ready to begin the actual process of mitosis. Mitosis consists of several phases, and the first phase of mitosis is called prophase. Pro, like in the word proceeding or coming first. So prophase is the first step of mitosis. So the chromosomes have already been duplicated. But what happens during prophase is the chromatin begins to condense, that is to coagulate and sort of separate out into truly visible chromosomes. So in this cell, you can see 4 chromosomes starting to separate out. Again, we have 46, but that would be too many to show in a figure. So they're showing you a cell that has 4, kind of like a fruit fly, I suppose. The other important piece of prophase is that those ropes that I mentioned earlier, the threads, begin to form. In the picture, they're called spindle fibers. So centrosomes, which are an organelle in the cell, are capable of producing microtubules. So the spindle fibers, the ropes that I've referred to, are actually microtubules. So think way back into the earlier part of the semester, we were talking about the cytoskeleton and microtubules. Well, here is another role that microtubules play in the cell. They're going to be the ropes that ultimately are going to pull these sister chromatids apart. So, to summarize, in prophase, the chromatin condenses into chromosomes, and the spindle fibers begin to form or originate at the centrosomes. The next phase of mitosis is called prometaphase. So what's happening here is the spindle fibers, the ropes, are latching on to the chromosomes. So you can see the 4 chromosomes there, each one existing as duplicate sister chromatids. The ropes have latched on and they're starting to pull on them just a little bit. So the other thing we see happening in prometaphase is the nuclear envelope, the membrane, the double membrane of the nucleus is breaking down. We're going to have to get the nuclear envelope out of the way so that we have room to pull these chromatids apart here in a second. And by the way, these spindle fibers, those microtubules, attach to the chromosomes at kinetochores. So another word that sounds like it starts with a C, it actually starts with a K, as you can see there. But spindle fibers attach to kinetochores on the sister chromatids. So that's prometaphase, the ropes attaching and nuclear envelope breaking down. The next step of mitosis is called metaphase. Meta literally means middle. You can think of it as middle because chronologically it occurs in the middle. But the other way to think about it is that all the chromosomes line up in the middle of the cell. So in metaphase, you can see that the spindle fibers have been tugging a little bit, and it's gotten all 4 chromosomes lined up single file along the equator or midline of the cell. So that's metaphase, all the chromosomes lined up single file along the middle. And then the spindle fibers will begin to shorten, and we will enter what's called anaphase. I think of those centrosomes that produce the spindle fibers almost like a fishing rod that has thrown out fishing lines, the fishing lines being the spindle fibers. So it's throwing out fishing line and it's caught a chromatid. And then those fishing reels start to reel in their fish. And if the centrosomes on opposite ends of the cell start shortening their spindle fibers, it literally pulls the sister chromatids apart. Technically, once they're apart, we don't refer to them as chromatids anymore. They're really only chromatids when they're held together. Now it would probably be more correct to refer to them each as chromosomes. So in anaphase, the sister chromatids separate and then move towards opposite poles. At this point, mitosis moves into the step called telophase and a process called cytokinesis. So telo means the end phase. And so we're getting to the end of the process. And you can see that the chromosomes have arrived at opposite poles, and we're beginning to form a new nuclear envelope around each set of chromosomes. And notice that each, what will become here a new daughter cell, has 4 chromosomes, just like the original cell had 4 chromosomes. We've just sent duplicate copies of each chromosome to each end of the cell. So in telophase, the chromosomes arrive, nuclear envelopes form around each set, so we'll get 2 nuclei in the cell, at least briefly. And then cytokinesis begins. Cytokinesis is the literal splitting in half of the cell. All these steps before it were just to get the chromosomes separated. But now we need to literally split the cell in half, and that process is called cytokinesis. And you can see the pinching in that's occurred there. The label there shows it as the cleavage furrow. So we can literally see the pinching in. And those little lines that you see in the middle of the cleavage furrow, those are actin filaments or microfilaments. So this is the cytoskeleton again contributing to a cell process. Those little microfilaments or actin filaments in the center are acting like a little noose or drawstring to pinch the cell in half. And eventually it will break into 2 separate cells. Here's a simpler figure of what's going on. You see the 2 purple nuclei have formed inside each new cell. And then the microfilaments are creating that noose or that ring that's going to end up pinching, creating the cleavage furrow, and boop, it splits into 2 daughter cells. So our one cell that started out in interphase has now gone through mitosis and divided into 2 daughter cells. And those cells are genetic clones of each other, and they're exactly like the original cell that started the process. So in fact, mitosis is used for growth and repair, basically when we need more identical cells. If you fall and cut yourself and scrape your skin open, you need more skin cells to fill that up to heal it, so we'll just use mitosis to just make more cells. Or for that matter, when you were an embryo growing into a fetus, that was all mitosis, growth and repair, just making more cells that are genetically the same as the previous cells. Here's an electron micrograph of the cleavage furrow. So here is an animal cell under the electron microscope, and you can literally see the pinching in that's being done by those microfilaments that will eventually pinch this into 2 separate cells. Now I've sort of been talking about human cells this whole time, but mitosis is also done by plant cells, for example. But plant cells do something a little different when you get to the cytokinesis part. In fact, plant cells do not do cytokinesis. They don't literally split into 2 because they have that rigid cell wall. You can see that this plant cell's got a rigid box forming its cell wall, and the cell wall is so stiff and rigid it will not pinch. So all the plant does is simply build a new wall right down the center. So you can see the new cell wall going up. It's getting a little bigger, getting a little longer, and eventually it will complete the cell wall and declare itself as 2 separate plant cells. So they don't do the cytokinesis pinching. They just literally build a new cell wall right down the center. Other than that, the mitosis is a very similar process. So let's look at some real cells and see if we can recognize the steps of mitosis. So what I'm showing you here is a cell right there in the center. You can see its nucleus, that little oval area in the middle of it. And this is a cell from a whitefish blastula. Now this particular cell is in interphase. I can tell because I look in the nucleus and I don't really see a lot going on in there. So the chromosomes are all tangled up as chromatin. Now recall that during interphase, that's really just normal cell activity. And during a blastula, that cell activity doesn't last very long because we need mitosis to happen pretty frequently. So at some point during interphase, the chromosomes will get replicated. Remember, the copying or the duplication of the chromosomes technically occurs during interphase. Once they've been copied, the cell can move into prophase. And recall what happens in prophase. In prophase, the chromatin condenses into visible chromosomes. So I'm starting to be able to see the individual chromosomes. In fact, chromosome literally means colored bodies. Chromo, color, soma, body. So chromosomes are colored bodies. So the tangled mess of duplicated chromosomes are now condensing so that we can sort of tell some of them apart. And the other thing that occurs in prophase is the microtubules begin to form at the centrosomes. The microtubules will be those ropes that are going to pull the duplicate sister chromatids apart. So then we move into prometaphase, and you can see the ropes have started to form and reach out there to grab the chromosomes. Each chromosome now exists as two duplicate chromatids, sister chromatids held together at a centromere. And the microtubules, the spindle fibers, attach to the chromatids at kinetochores. So the fishing lines have been thrown out, and the hook, call the hook the kinetochore, I guess if you want, the hook has attached to the chromatids. And the nuclear envelope has broken down because we need to get all that nuclear membrane out of the way so we can get these chromosomes lined up and pulled apart. So the nuclear envelope breaks down during prometaphase. Then in metaphase, everything is lined up in the center. The spindle fibers, those pink stringy lines that you can see going out to the middle, have latched on and they're starting to tug at each other a little bit, and it's drawn everything into the center during metaphase. Then those fishing reels will reel their microtubules in and we'll see things move apart. But metaphase is when the chromosomes line up single file along the equator of the cell. So now let's start shortening those microtubules, and we move into anaphase, and you can see the sister chromatids, the duplicate copies of a chromosome, have separated. They're being pulled towards opposite ends of the cell. That means each end of the cell is getting a set of chromosomes, and they're identical sets going to each end of the cell. So microtubules shorten, and the sister chromatids separate and move towards opposite poles. And again, technically, once the sister chromatids have separated, they get to be referred to as chromosomes in their own right as they move towards these opposite poles. That moves us on into telophase, the end phase, and you can see here that the chromosomes have arrived and we're forming a new nucleus around each end. So the chromatids, now called chromosomes, arrive at opposite poles, and a nuclear envelope forms around each set of chromosomes. And then, once that's happening, or actually sort of while that's happening, cytokinesis begins so that the cell can literally begin to split into the two daughter cells. So that's mitosis in an animal cell. I did mention that this goes on in plants. So here's an interesting picture from under a microscope. This is the root tip of an onion. So roots, as you know, are constantly growing out into the soil where they can absorb nutrients. So the tips of roots are always undergoing mitosis as the root lengthens and elongates. So if you put a little slice of onion root tip under the microscope, you can catch cells in various stages of mitosis. So this is a snapshot, and if you just look, you can see a lot of interesting stuff going on inside the nuclei of these cells. So here, for example, looks like a cell that is in prophase. The chromatin is condensing into visible chromosomes, but I can tell the nuclear envelope is still intact. So that's prophase. Whereas this one is prometaphase. You can see that the nuclear envelope is beginning to break down, but I can still see the chromosomes. This cell, it's probably the best case for one that might be in metaphase under the microscope here, where the chromosomes have lined up in the middle. And then here is clearly anaphase. You can see the sister chromatids have separated and now called chromosomes are moving towards opposite poles. And then finally, this is one that looks like it's in telophase because they've arrived at opposite poles, starting to build a new nuclear envelope around each set. And you can even see that cell wall forming right down the center that's going to eventually separate this into two separate cells. Alright, so that's mitosis. That's our first video lecture on cell division, so part 1. In our next video, part 2, we're going to look at how that process of mitosis is regulated and also what can happen if it becomes unregulated. And that's a pretty interesting story. So we'll talk about that in the next video.